The purpose of this study was to expand the scientific community's understanding of how secondary peptide structures formed from amino acid mixtures on the prebiotic Earth. This study focused on which peptide structures were formed from mixtures containing many different monomers, while previous studies simply focused on the products of one or two monomers. Also, instead of creating sets of monomers from known secondary peptide structures, the study constructed new secondary structures from monomers.
An important component of the study involved understanding depsipeptides. In order to define depsipeptides, one must first understand peptides. Peptides are chains of amino acids. When joined in long chains, they form polypeptides, which ultimately become proteins. Depsipeptides have at least one ester bond and one amide bond. The study explained how depsipeptides likely form during common processes such as evaporation and rehydration, rather than during a few isolated dramatic changes in Earth's atmosphere. In the lab, researchers mimicked early atmospheric conditions on Earth by hydrating and drying the depsipeptides several times. Additionally, they are able to form rapidly in a laboratory setting when exposed to circumstances similar to those that would have been present on prebiotic Earth. The creation of depsipeptides in the lab supports the idea that modern peptides are likely formed from various simple molecules that become more complex over time. The researchers studied a potential pathway for the formation of peptides based on reactions containing ester and amide bonds. Ester linkages are considered to be more stable, however they are more susceptible to hydrolysis. In contrast, amide linkages are more stable during hydrolysis. Thus, the prevalence of amide bonds increased over a few water evaporation and rehydration cycles.
The data showed that glycolic acid and lactic acid were more likely to be present in the depsipeptides than malic acid. This result contradicted the hypothesis because the researchers thought that malic acid would be more prevalent in the depsipeptides. Only 3 amino acids and 3 hydroxy acids were tested in the study because of the large number of possibilities of combinations. Using more amino acids and hydroxy acids would have made the data too difficult to analyze.
The diagram on the screen shows the different types of depsipeptides made by each solution. Each orb corresponds to a different depsipeptide, with larger orbs corresponding to the more prevalent species, and darker orbs signifying a larger percentage of amino acids present in the depsipeptide. The orbs are placed in 3D space according to their mass, charge, drift rate, and retention rate. Differentiating depsipeptides based on 3 criteria helped represent the diversity present in the solution. All 3 graphs show a positive correlation between mass and drift time. More importantly, the graphs show the comparative rarity of depsipeptides formed in the malic acid solution. Although the lactic acid seemed to form more depsipeptides than the glycolic acid, the lactic acid and glycolic acid solutions have similar numbers of orbs.
The study provided further insight into the rise of organic molecules and building blocks of life that could have arisen from prebiotic Earth. Most studies on the diversity of protopeptide sequences have been mathematical or top-down, looking at current sequences and seeing where they could have come from. This study provided a bottom-up approach, seeing what sequences formed from likely pre-life Earth conditions. The study demonstrated the usefulness of considering not just amino acids that code for existing proteins, but all of them that might have been present in prebiotic Earth. This experiment found several that could have possibly catalyzed the length increase of later and currently observable peptides. This paper relates to Module 1 because it involves the study of evolution and how macromolecules arose from inorganic environments and assembled to the molecules that we see today. Because of how they assemble, this relates to Module 3, as these protopeptides led to the assembly of modern-day amino acids that make proteins.